Hi, my name is Gabriela Barrera, and I'm Group 1, and we have role of glycoproteins in cell recognition. Glycoproteins are found on the extracellular matrix of the plasma membrane, which is the surface of the cell, and can be defined as a protein embedded in the phospholipid bilayer with a carbohydrate chain attached to it on the outer surface of the cell, as you can see on this image. Glycoproteins exist in cells, mostly all over our body, and they play a role in the structure, reproduction system, immune system, hormones, and protection from degradation. And the word glycoproteins comes from the prefix glyco, which means proteins, and since it's combined or binded to a protein, they form the word glycoproteins. The structure of glycoproteins can vary based on the shape that the carbohydrate chains obtain. These carbohydrates are made of oligosaccharides that can be straight or branched and may consist of about 2 to 60 sugar units per chain, and some of these sugar units are shown below this bulletin. These sugars are covalently bonded to the protein that's within the phospholipid bilayer of the membrane. Also, once the proteins are secreted and glycosylated, they will form N-linked or O-linked sugars, which you can see on the image to the left. And what differentiates them is that they are linked to an oxygen, the O-linked sugars, and the N-linked are sugars linked to a nitrogen. Cells use cell adhesion molecules, also known as transmembrane proteins, and carbohydrates that act as strong antigens as a type of cell recognition. This process uses glycoproteins as receptors to bind a cell with another cell. Glycoproteins are found floating inside or around the membrane of cells, as you can see on the image below. They can be found across all living organisms, and one example of glycoproteins in our body is mucins, which are secreted in the mucus of the respiratory and digestive tracts. The sugars that make up the glycoproteins are able to attach to two parts of the cells, which are the ER and Golgi apparatus, and if they attach to the Golgi, then they will produce the O-linked sugars as mentioned on the previous slide, and if they are attached to the ER, then they will produce the N-linked sugars. Glycoprotein recognition function is to mainly protect the cell from becoming attached or adhered to pathogens such as virus, bacteria, and toxins, while also allowing the cell to have heterophilic and homophilic interactions with other cells through the binding of adhesion molecules. They also help regulate the behavior of cells in multicellular organisms. Some diseases associated with glycoproteins and a malfunction in them is what is known as the lysosomal storage disorder, which occurs when the enzymes within the lysosome do not function to their full potential or are inactive, causing the accumulation of sugars or substrates that are regularly broken down and degraded. Causes of these storage disorders with specific sugars cause the diseases, which include amyloidosis, fucosidosis, sialidosis, and Schindler's disease, as well as other diseases. Glycoproteins have also been found to have a role in this, the protein misfolding through the addition or modification of the N-glycans in the ER or the Golgi apparatus. A recent study suggests that addition and modification of the N-glycan core is intimately linked to the folding, disulfide bond formation, and complex assemblies of glycoproteins. Just like the lysosomal storage disorder, there is such thing called the ER storage disease or disorder, which is the accumulation of misfolded proteins in the ER, and the N-linked glycans play a major role in determining whether or not the protein is giving access to the Golgi or if it becomes degraded in the ER. If the ER allows a misfolded protein to go through to the Golgi, then the Golgi will detect the N-glycans misfolding and degrade it completely. And you can see, get an idea of how that process is on figure 1. And if the misfolded proteins remain in the ER, then they are more prone to becoming aggregated, as well as to them being soluble or insoluble. Based on their solubility, their degradation process is slightly different from figure 1, and you can see the two types of processes they take in, if they're soluble or not. I was having a little bit of difficulty trying to find information regarding this topic. So my question is, why is there more information regarding the adhesion of glycoproteins and the cell recognition of glycoproteins? And is this because they go hand in hand with how they are able to function?